Good morning, everyone. Thanks to the organizers for the great opportunity to present our work today, the relationship between HLA-B 0702, HLA-C 0702 alleles, frailty, and the cellular immune response to cytomegalovirus. So instead of like epigenetics, my study is actually focused on the genes and the cellular response. Studies have shown that frailty is associated with chronic low-level immune activation and inflammation. One of the potential drivers of such immune activation is persistent infection with cytomegalovirus, CMV. In men living with and without HIV in the multi-center AIDS cohort study, MAX, we found that CD8 T-cell responses to CMV were significantly and strongly associated with serum concentrations of some inflammatory markers. For example, TNF-alpha, IL-8, monocyte chemoattractant protein MCP1, and MCP4. These CD8 T-cell responses also predicted onset of frailty. To trigger CD8 T-cell responses, antigen presentation by human leukocyte antigen class 1 moleculars is required. And genes encoding the HLA class 1 moleculars are highly polymorphic. There are 3 loci of HLA class 1 genes, A, B, and C, and each locus has multiple alleles. Therefore, we started the association between HLA class 1 alleles, CD8 T-cell responses to CMV, and frailty in the MAX. The study included 40 men from the Baltimore, DC side of MAX, 21 with virologically suppressed HIV, and 19 without HIV. Frailty was assessed by frailty phenotype. Among 40 men, 21 were non-frail and 19 were frail. CD8 T-cell responses to CMV was assessed by flow cytometric analysis of production of IF and gamma, TNF-alpha, and IL-2 in response to overlapping peptide pools spanning 19 CMV open reading frames. A positive response was defined as equal to or larger than 0.05% of the cells producing any cytokine after background subtracted. And the HLA-ABC was genotyped by PCR-SOAP, sequence-specific oligonucleotide probing, and the data was retrieved from the database of MAX. Fisher's exact test was used to evaluate the significance of association of HLA-1 alleles with frailty and with CD8 responses to CMV. And significance level was defined as p-value of less than 0.05. We did not adjust for multiple comparisons because of the small sample size. And first, we examined the association between HLA class 1 alleles and frailty. We found that HLA-B alleles differed significantly by frailty status, but HLA-A and HLA-C did not. And the difference in HLA-B alleles was due to expression of HLA-B 0702. As shown in this table, among all men, 12 expressed B0702 and 28 expressed alleles other than 0702. And the expression of this HLA-B 0702 was more prevalent in non-frail men than in frail men, with an odds ratio of expression of this allele in non-frail men versus frail men equaling to 7.7. And we also did not see any significant difference of this allele expression between HIV negative and HIV positive men. And next, we examined if frequency of CD8 responses to any CMV ORFs differed by frailty status. So in this graph, the y-axis represents the percent of men responding to peptide pools spanning a given CMV ORF, and the x-axis lists all the CMV ORFs that we tested. And on the left is UL83, which elicits the most frequent response, and then on the right is UL94, which elicits the least frequent CD8 response. And then the blank bar represents non-frail men, and dotted bar represents frail men. So as shown in the blue circle, the CD8 T-cell responses to UL99 and to UL28 was significantly higher in non-frail men than in frail men. For UL99, 71% of non-frail men responded, and for frail men, only 33% responded. And then for UL28, 48% of non-frail men versus 10% of frail men responded. And interestingly, we found the response to these two CMV ORFs was significantly associated with expression of HLA-B 0702. For UL99 specific CD8 T-cell response, half of the positive response was associated with expression of 0702. And for UL28 specific CD8 response, the association was much stronger, with almost all of the positive responses was associated with expression of 0702. After searching the literature, we found that peptide derived from UL28 triggered strong HLA-C 0702 restricted CD8 T-cell responses. But HLA-B 0702 was reported to be in linkage disequilibrium with HLA-C 0702. And this is true in our study group. The expression of these two alleles is highly concordant, with only one exception. And because of this linkage disequilibrium between HLA-B 0702 and HLA-C 0702, HLA-C 0702 was also significantly associated with the T-cell response to UL28 and UL99 and with frailty, which are not shown here. To summarize, we found that expression of HLA-B 0702 and HLA-C 0702 alleles was significantly associated with a lower prevalence of frailty in a small cohort. Thus, these alleles may be protective against frailty in both HIV-negative and HIV-positive men. Because these alleles was also significantly associated with stronger CD8 T-cell responses to peptides spanning CMV ORFs UL28 and UL99, it is possible that the HLA-associated protection against frailty is mediated by the T-cell response to CMV. The limitations of the studies are, first, the study is cross-sectional, and second, the sample size is small. This hypothesis and the limitations will be addressed using data on HLA and frailty from the entire MAX. Finally, I'd like to thank our collaborators at Oregon Health and Science University, Dr. Picker and Dr. Sylvester, for their provision of most of the peptide pools, and participants and the staff of the MAX for their dedication to the study, and the funding agencies for supporting the study. Thank you.
Yeah.